Welcome back to today's podcast. Today we are talking about a major question in neonatal nutrition. What happens when a baby is born before the body is metabolically ready for life outside the womb? Preterm birth is not simply early delivery, it interrupts a critical period of growth, nutrient storage, intestinal maturation, brain development, and metabolic programming. During the third trimester, the fetus normally receives a continuous supply of nutrients from the placenta, but when an infant is born very preterm, that placental supply suddenly stops. At the same time, the infant now has to breathe, regulate body temperature, maintain circulation, digest nutrients, grow, and support rapid brain development. That creates a major mismatch: high energy demand, but immature metabolic capacity. Why nutrition is so important in very preterm infants. Very preterm infants are born before they have completed normal nutrient accretion. This means they often begin life with nutritional deficits that need to be corrected after birth. The paper we are discussing focuses on enteral nutrition, nutrition delivered through the gut, and why it is so important for very preterm infants. Historically, clinicians were cautious about feeding preterm infants early because of concerns about feeding intolerance, intestinal injury, or necrotizing enterocolitis. But the field has shifted. Current evidence supports early and consistent enteral feeding, especially using mother's own milk when available. The key point is this: enteral nutrition is not just calories, it is a developmental signal. When nutrients enter the gut, they stimulate intestinal growth, improve gut barrier function, promote hormone secretion, shape the microbiome, and may influence immune and brain development. The developing brain has huge energy needs. One of the most important ideas in this paper is that the developing brain is extremely metabolically demanding. In fetal life, the placenta supports this energy demand, but after preterm birth, the infant loses that continuous nutrient supply. At the same time, the brain is still undergoing rapid white matter development, neuronal growth, and myelination. So nutrition in the early postnatal period becomes a major determinant of brain growth and neurodevelopmental outcome. This is why the first days and weeks after preterm birth are so important. Poor postnatal growth is associated with poor brain growth and worse neurodevelopmental outcomes later in life. Why human milk matters. The review strongly emphasizes mother's own milk as the preferred source of enteral nutrition. Mother's milk provides nutrients, but it also contains bioactive components that formula cannot fully reproduce. These include immunoglobulins, human milk oligosaccharides, lactoferrin, extracellular vesicles, microRNAs, cytokines, and other factors that support gut maturation and immune regulation. Colostrum, the first milk produced after delivery, is especially rich in immune and bioactive compounds. It may help reduce inflammation, support intestinal development, and improve feeding tolerance. When mother's own milk is not available, pasteurized donor human milk is generally preferred over formula because it lowers the risk of necrotizing enterocolitis. However, donor milk is not identical to mother's own milk. Pasteurization reduces some bioactive components, and donor milk may contain less protein and some minerals. So the hierarchy is usually mother's own milk first, donor human milk second, preterm formula when human milk is not available or when risk is lower. Why fortification is necessary. Even though human milk is biologically ideal, unfortified human milk usually does not meet the nutritional needs of very preterm infants. Very preterm infants need high amounts of protein, energy, calcium, phosphorus, iron, zinc, and vitamin D to support growth and development. That is why fortification is essential. Fortifiers add nutrients to human milk so that preterm infants can meet growth targets. The paper discusses standard fortification, adjustable fortification, and targeted fortification. Standard fortification is easier, but it assumes average milk composition. Targeted fortification is more precise because it measures the nutrient composition of milk and adjusts supplementation accordingly, but it is more expensive and resource intensive. The central issue is balance. How do we preserve the benefits of human milk while still meeting the high nutrient requirements of very preterm infants? Where fat metabolism comes in. This is where our bigger question begins. Preterm infants receive lipids as part of parenteral and enteral nutrition. Lipids are essential because they provide energy, support growth, and supply fatty acids needed for brain development and myelination. But the question is, can preterm infants efficiently handle and oxidize these lipid substrates? Fat metabolism depends on several pathways. First, mitochondrial beta oxidation breaks down fatty acids to generate acetyl coenzyme A, NADH, and FADH2, supporting ATP production. Second, peroxisomal oxidation helps process very long-chain fatty acids and complex lipid substrates. Third, omega oxidation can become more active when mitochondrial beta oxidation is limited, producing dicarboxylic acids. If these pathways are immature or overwhelmed, lipid handling may become inefficient. Instead of being fully oxidized for energy, fatty acids may accumulate as triglycerides, acyl carnitines, dicarboxylic acids, or other partially oxidized intermediates. This could contribute to hepatic lipid accumulation and broader metabolic stress. What happens when fat oxidation is not optimal? If preterm infants cannot efficiently oxidize fatty acids, the body may compensate by relying more heavily on other fuels. That means increased use of carbohydrates and amino acids. Carbohydrates may be used to support glycolysis and maintain blood glucose. Amino acids may be catabolized to provide carbon skeletons for the TCA cycle or gluconeogenesis. But this compensation has costs. Greater amino acid catabolism can increase nitrogen disposal burden. Altered carbohydrate use can affect insulin and glucose handling. Incomplete fatty acid oxidation can alter TCA cycle flux and mitochondrial redox balance. So impaired lipid oxidation may not be an isolated lipid problem. It may shift the entire metabolic network. This creates the concept of metabolic inflexibility in prematurity. The preterm infant may have nutrients available, but may not have fully mature metabolic machinery to use those nutrients efficiently. Why this matters clinically? This matters because neonatal nutrition is often designed around nutrient delivery. How much protein, how much lipid, how much energy, how fast should feeds advance, what type of milk or fortifier should be used. But an equally important question is, how does the preterm infant metabolize the nutrients we deliver? Two infants could receive the same amount of lipid, but if one has lower mitochondrial oxidative capacity, that infant may accumulate more lipid intermediates or shift toward amino acid catabolism. This is why future studies need to combine nutrition trials with metabolic phenotyping. For example, we need to measure acyl carnitines, dicarboxylic acids, ketone bodies, TCA cycle intermediates, amino acid catabolism markers, glucose and insulin responses, hepatic lipid accumulation, mitochondrial function. This would help us move from simply asking, what should we feed, to asking, how is the preterm infant using what we feed? Closing, the main takeaway from this paper is that early, consistent, human milk-based enteral nutrition is central to improving growth, gut development, and neurodevelopmental outcomes in very preterm infants. But this also opens a deeper metabolic question. Preterm infants are exposed to high nutritional demands during a period of immature hepatic, intestinal, and mitochondrial development. If fatty acid oxidation pathways are not fully mature, lipid delivery may interact with amino acid and carbohydrate metabolism in ways that shape growth, energy balance, and hepatic lipid accumulation. So the next frontier is not only optimizing nutrient delivery, it is understanding metabolic capacity. Because in preterm infants, the question is not just how much nutrition do we provide? The question is also, can the infant metabolically handle and use that nutrition efficiently? Thanks for listening.